Hello everybody, my name is Kyle Alexander. This is my presentation for Bio 1010 and the topic is bacteriophages, viruses that prey on bacteria. These are very incredible organisms, or I should say less than. They are indeed viruses and not even considered life, but they're one of the most numerous biological machines that we have on Earth. So let's go ahead and begin. So, introduction, when it comes to viruses, what are they? They're non-living biological machines that require a host cell to replicate. So they have no means to self-replicate, but they require a host cell to replicate. Their genetic material is enclosed in a capsid. It's often referred to as a head as well. It can either have DNA or RNA, but not both. It can just have one or the other. They're all considered non-living because they do not use energy and they're not made of cells. And this specific group that we're talking about today is a virus that only infects specifically bacteria. Bacteriophage are viruses that infect and destroy bacteria, and the name comes from Greek where it means bacteria eaters. Their structure consists of a head or capsid, tail sheath, base plate, pins, and tail fibers. As an image on the left here of what it looks like, and it's an extremely small, a very small virus that can only be seen by an electron microscope. And when it comes to a life cycle and reproduction or replication, it has two strategies that it utilizes. The first is the lytic life cycle. As you can see in the image on the left, the phage then adheres to a bacterial host cell that it has very specific tail fibers that it can actually adhere to the proteins on the outside of the bacteria. After that, it penetrates its viral DNA into the cell, or it's the bacteria. At this time, biosynthesis occurs, phage DNA replicates, and phage proteins are made. So it's hijacking the bacterial mechanisms or machinery inside to create its own phage parts. And then once those parts maturate, or during the maturation phase, new phage particles are assembled, so it's all brought together. And then at the fifth cycle, the lysis, so the cell lyses and releases all the phage from the cell, and the cell implodes and dies. And so this is a short-term strategy. It can happen in the matter of hours, and it is the fastest way to make as many phages as possible at the expense of the host cell. So it's a bit like going in guns blazing, versus the second life cycle is a bit more methodical. This is the lysogenic cycle, and the lysogenic cycle is a little bit different because it's a little bit delayed on when the bacteriophages are released. It absorbs its DNA, so it penetrates through the cell membrane or cell wall, injects its own DNA or RNA. That RNA and DNA then become integrated with the host cell's DNA or RNA, and then from that, the cell is going to continue its life cycle. So it can continue to divide. The only thing that's been changed to this cell is its genetic structure has been intertwined with the bacteriophage, and at some point there's going to be a tipping point where then the cell is going to then produce parts for phage and eventually lyse. But the cell continues to replicate, and what makes this strategy so effective is that all the offspring that have been infected, or that are related to the original infected host, are also going to produce phage. Induction, when the daughter cell is induced, the viral DNA is excised, so the viral DNA then leaves the genome of the bacteria. Then it synthesizes proteins and creates more parts for the phage. At this point, all the parts are brought back together in assembly and then lysed and released, and this can happen on a huge scale, which is absolutely devastating to the bacterial population. This is a more long-term strategy that is harder for these bacteria to quickly adapt to, because it's the infection is passed down, and it's more of a covert stealth agent approach. When it comes to genetic material and replication, regardless of which cycle is being used, bacteriophage hijack their host to replicate. They use the host's mechanisms of cellular processes to create more of themselves rather than them doing it themselves. And so it's accomplished by the bacterial ribosomes and enzymes who replicate. And they use a central dogma of biology, which is the viral DNA has been injected into the bacteria. So it goes from DNA to transcribed to RNA, and then that translates into proteins, which then are parts for the phage. The bacteriophage are extremely discriminatory. They only go for very specific strains of specific species of bacteria, and they also don't really interact with anything else, especially to humans, which is something that's potentially beneficial to future therapies. While these two forces are engaged in this perpetual battle for existence, they're constantly microevolving to combat each other's strategies. Microevolution is used on both sides of the conflict to combat adaptations. Bacteria evolve to change their surface structure to keep phage from attaching to them, while phage also evolve by acquiring new receptors and acquiring new genes from previous bacteria that they've previously hijacked. Their scale of bacteriophage, where they're absolutely everywhere. They're responsible for killing 40% of the bacteria in the ocean every single day. There are more bacteriophage on Earth than any other organism combined, including bacteria. And there's an estimated amount of 10 to the 31 power on Earth, which, a side note, there are more bacteriophage than there are stars in the observable universe. There's estimated to be 1 trillion bacteriophage for every single grain of sand on the Earth. Absolutely massive amounts. And they're found everywhere. They exist everywhere where living things exist. Within the biosphere, they're the most numerous biological entity. They're in our ocean, they're in our soil, and even within our bodies. So, with that being said, there are very real possibilities that phage can be used as a therapeutic alternative to antibiotics. Since we have been going through these many generations of antibiotics, bacteria have increased their resistance to antibiotics. And we've been using phage to help turn the tide of antibiotic resistance because antibiotic resistant bacteria are vulnerable to bacteriophage. And so when they become resistant antibiotics, they lose their resistance tolerance to bacteriophage, putting these superbugs like MRSA in a catch-22 where they're highly susceptible to phage, but they are very hardened against antibiotics. And so we can introduce both of them to hopefully eradicate these kind of superbugs that have been plaguing hospitals and various other sectors of healthcare. In summary, bacteriophages are viruses that infect bacteria to reproduce. They use a lytic and lysogenic life cycle. Bacteriophage use the dogma of biology and the reproduction cycle, and they also prove to be promising for medical breakthroughs for treating bacterial infections. And these are all my resources, and I would just want to thank you again so much for watching this video. And please give me some feedback in areas that I can improve. Thank you so much.